said before, I already explained this side of the mnemonic regarding triglycerides. Now I'm going to explain this side of the mnemonic regarding cholesterol. So if you remember, I described cholesterol's structure as being a ring structure, like in chemistry, what we call the structure. Okay, somebody said if we can go over APO 100 at the end, I'm totally okay with that. At the end, I'll go over ApoB 100, and we'll go over and make sure everybody remembers every single apolipoprotein, because for sure, that's important. But yeah, back to this mnemonic. For cholesterol, it has a ring structure. That's what we call this structure in chemistry. And so the mnemonic I want you guys to remember is associated with like an engagement ring, right? And the reason I want you guys to remember an engagement ring is that I sort of want you to remember this image in your head, which is like a proposal. And the reason I want you to remember that is because cholesterol is associated with steroid synthesis. Steroids are certain like fat-based hormones in our body, and specific examples of steroids are stuff like estrogen, testosterone. So that's why I put an image of a proposal over here, because maybe you'll remember, like a guy and a girl, and it will make you remember, oh, estrogen and testosterone. Maybe you can imagine the guy as being really buff or something, so he had to take steroids, for example, to get the girl, like on The Bachelor or something. I also want you guys to know how like there's this really big heart in the background. And it's because if you remember, I talked about a very, very specific complication. It's very high yield to know for cholesterol, which it doesn't involve the heart, but involves blood vessels, which eventually can lead to effects in the heart, such as a myocardial infarction, which is a heart attack. Okay. So I will explain the mechanism later, but what I want you to know for this mnemonic is that cholesterol can have effects on blood vessels, right? Specifically, it's going to harden the blood vessels, and that can lead to effects on the heart, okay? Cholesterol is also involved in steroid hormone synthesis. Examples of steroid hormones are like estrogen, so estrogen for the girl, testosterone for the boy. Even if you wanted to remember cortisone is our stress hormone. It goes up probably right now for you when you're near exams. So maybe, his heart's beating really fast because he has a lot of cortisone because he's very stressed. And that's also a steroid hormone. And the last thing is he proposed on a very sunny day. And that's because when I think of the sun, I think of vitamin D and cholesterol. If you guys remember the vitamin D pathway, I think you guys learned it in GI or something. Cholesterol is needed to make vitamin D. So that's the things that I want you to think of when you think of cholesterol. One other thing I really want to point out right now is that for triglycerides, I really, really emphasized in your guys' head that like chylomicrons have the highest percentage of triglycerides, and then the second most is VLDL. For cholesterol, especially when I'm talking about bad cholesterol, like too much cholesterol, mostly it's LDL. Anytime I'm going to talk about too high cholesterol, I'm mostly referencing LDL, because we talked about before how HDL is good for us. So when I talk about disorders and when I talk about the bad effects of cholesterol, I'm mostly going to be referring to LDL. Like I said before, nobody cares about IDL, so don't worry too much about that. The other thing I want you to remember with this mnemonic of a ring is that this ring circular structure, right, with a very stable stone in the middle, should remind you that the function of cholesterol is actually that it exists in between the phospholipid bilayer of our cell in order to stabilize it. So if you can imagine, I have this phospholipid bilayer, and these two layers, this bottom layer over here, and this top layer over here, they would just constantly be sliding against each other if I didn't have anything to stabilize it. And so my cell wouldn't be able to stay in a circle. It would just be sliding around everywhere. But fortunately, I have little free cholesterol that's in my cell membrane, and it stabilizes my cell membrane. Okay, so remember I said that fats have a structural purpose. Specifically, what I'm referring to when I say that, I'm saying that cholesterol serves to stabilize my cell membrane. Okay, and hopefully maybe that looks a little bit similar to this ring over here and how there's a very stable rock in the middle. So that's the mnemonic I have for cholesterol, and we're going to go into the details and explain each of those mechanisms. Okay, so just to recap from the previous part, because I'm going to go into more details. If you remember in the previous lecture, I told you that inside of my liver, I have cholesterol inside of my liver, right? And when I eat a really fatty food, what's going to happen is that the cholesterol is going to be added to my bile or my bile salts or whatever, and is going to enter into my GI tract so that the food that I just ate, the really fatty food, can be broken down from my bile salts or from my bile salts. And then after I've sort of like digested that meal or whatever, the cholesterol needs to be reabsorbed from my chylomicron, okay, so that the cholesterol can go back to my liver. Okay, so that's sort of like how cholesterol is recycled. Don't let this confuse you. Yes, there's a small amount of cholesterol in chylomicrons, but the main role of chylomicrons is still delivering triglycerides. But I just wanted to show you guys how cholesterol is being delivered. We also talked about how the liver can make these three different lipoproteins. It can make VLDL, IDL, or LDL. If I say this in a different order, it would be LDL, IDL, VLDL, LIV. LIV stands for liver because these three are the ones that are made from the liver. These are the three lipoproteins made from the liver. Because they are made from the liver is going to give them ApoB100, right? It's going to give each of them, or it's going to give the VLDL ApoB100, and the liver, the function of the ApoB100 is so that once it's attached to the lipoprotein, now the lipoprotein can go to other cells in my body, not my liver, but my other cells in my body, in order to deliver the lipids to the peripheral tissue for energy or whatever. Just as a recap, VLDL delivers triglycerides, and something like LDL delivers cholesterol in general. And so yeah, that's why these three have ApoB100, because they all come from the liver, and the liver is telling them be 100% prepared for delivery, so that when they deliver, they still have it. And I also told you that my HDL is my angel, it's my hero. We like this. The cholesterol is considered the good lipoprotein, because it is the only one that is going to carry the cholesterol from my peripheral tissue, and by the ways, from my peripheral tissue and in my blood. The cholesterol that's been stuck in my blood that I'm going to show you later on is going to be delivered back into my liver through HDL. Okay, so HDL is the only one that does reverse cholesterol transport. It goes from my peripheral tissues back into my liver. Okay, and it's HDL is the only one that has ApoA, the capital A, ApoA lipoproteins. And it's the only one that interacts with the LCAT enzyme. If you guys remember the mnemonic, it was like Hadil always gets A's. So she's the one with the apolipoprotein A. And she's the only one who passed her law college admissions test, LCAT, because HDL is the only one that interacts with LCAT, because the role of LCAT is to take the cholesterol that's in my peripheral tissue, make it cholesterol ester so that it can enter into my HDL, so that now the cholesterol ester is inside of my HDL can bring it back into my liver, okay? So that's sort of like a recap. Let's just briefly go over the other ones actually right now. ApoB48 is only found on chylomicrons, 8 for 8 food. ApoB100 already explained. ApoC2, as you can see, it's only found on chylomicrons and VLDL, because the function of ApoC2 is that it activates lipoprotein lipase, which is found like in your adipose tissue, in your muscle cells, on your blood vessel walls. And the function of lipoprotein lipase is that it breaks, or it cleaves, or it cuts triglyceride into 2 parts. So cleaves triglycerides into 2 parts. Should make you think of ApoC2. And because chylomicrons and VLDL are the ones that are responsible for carrying triglycerides, those are the ones that carry ApoC2, because these are the ones that need to activate LPL. The rest of the ones don't really play a role in triglyceride breakdown, not as much at least. However, I did tell you that IDL and HDL do have a little bit of triglycerides, and the triglycerides that are found in IDL and HDL can be broken down by another enzyme called hepatic lipase. Does the same thing as lipoprotein lipase, except it's found in the liver, okay? The other lipoprotein lipase is found anywhere else in the body but the liver. Hepatic lipase is only found in the liver. And hepatic lipase breaks down the triglycerides in IDL and HDL, okay? And then lastly, we have ApoE. If you look at ApoE, just remember that it's on everyone else but LDL. And the reason that exists is because the LDL can directly go to the liver. Okay, the LDL receptor that's located on a hepatocyte will automatically recognize the LDL. There's no extra apolipoprotein that's needed. However, for every single one of these, the other ones, okay, they all need this extra ligand, this extra protein called ApoE, because the hepatocyte will not recognize these other lipoproteins without ApoE, because the receptor on the hepatocyte was designed to only recognize LDL, like just by nature. It only recognizes LDL. So all of the other ones, they need ApoE, and that will make the lipoprotein sort of disguise itself as LDL, and therefore the LDL receptor that's on the hepatocyte will be able to recognize these lipoproteins, chylomicron, VLDL, IDL, and take in those lipoproteins. So that's just a recap of everything that we've covered so far. And maybe it makes a little bit more sense of why I have like how to categorize these and how to memorize these. Okay, so as I'm gonna quiz you guys now. So I told you that, well, I'm gonna ask you like the bile acids and the bile salts, what are they made of? What kind of fat are they made of that gets secreted by the liver? Is it triglycerides or is it cholesterol? Yeah, okay, I see some answers. Okay, good. You guys said cholesterol, that's correct. Okay, and then I already told you guys this before, but I told you that the role of the role of the LDL is so that it delivers the cholesterol to peripheral tissues in order, if they need to like make, for example, a steroid hormone, then the cholesterol is going to be delivered to those peripheral tissues. And I gave you some examples of some steroid hormones as well. And then what part of the cell? So I told you there's a very specific part of the cell that requires the cholesterol for the cholesterol function. Yeah, plasma membrane, exactly. Okay, and I kind of already gave you the answer to this. I said it 2 seconds ago, but there's some other tissues that require cholesterol for what kind of hormone? What specific kind of hormone am I talking about? Yes, steroid synthesis. Yeah, if you remember my mnemonic, I gave the proposal as an example. Okay, and in the previous lecture, if you remember, I said that there was only one apolipoprotein that is found on LDL. It's only one apolipoprotein that's found on LDL. Yeah, okay, I see one answer. Does anybody else want to answer as well? Okay, yeah, it is ApoB100. ApoB100 is the only lipoprotein that is found on LDL. And obviously, ApoB48, I told you, is on chylomicrons, not on LDL. Okay, so I'm going to explain to you the steps of how, and so this is my LDL. I told you that its role was to deliver cholesterol to peripheral cells, right? So for example, maybe like the testes, they need to make testosterone. And if they need to make testosterone, they need cholesterol. So that's just an example. So I have my LDL and it needs to deliver cholesterol to this cell over here. And I'm going to explain to you the steps on it. And then I'm going to quiz you on it in the next slide. So LDL, I said it only has one apolipoprotein. It's apolipoprotein B100. The apolipoprotein B100 is going to the function of it is so that it attaches to a receptor on the cell surface. And once that binding happens, once the ApoB100 attaches to the receptor, it's going to be endocytosed. It's going to be taken into the cell. What you have to remember is this protein over here called clathrin. And the role of clathrin is that it like stabilizes this pit over here. And so it's sort of involved in like the first step of endocytosis. So clathrin is a protein that's found on the inside of the cell, okay, on the inside of the cell. And its role is to sort of like initiate endocytosis, right? Like it makes this little pit so that eventually this pit, once it takes in the LDL, will form like a circle. And we call that full circle a vesicle. Okay, so remember clathrin protein, clathrin. Okay, so once the LDL is taken in, it's taken into this vesicle, and we call this vesicle an endosome. And then the endosome is actually going to like fuse, or it's going to like find lysosomes. And I don't know if you guys learned this in high school, but the function of lysosomes is to literally break down stuff inside of the cell. So what's happening is that after the LDL enters into my endosome and then the lysosomes get added, the LDL will be broken down. Like remember I told you there's like an inside part and an outside part. That outside part is going to be broken down. That phospholipid monolayer is going to be broken down. And now that I've broken down my LDL, I have free cholesterol, right? So my lysosome broke down my lipoprotein, broke down the LDL, and now I have free cholesterol that is inside of the cell. Another thing I really want to emphasize to you is that if you look at this, the receptors are being recycled, okay? So after the lysosome breaks down LDL, the receptor that was responsible for first attaching to LDL will just go back onto the surface so that the next time another LDL, comes into the bloodstream, it can attach again. So the cycle is just going to repeat. So the idea here that I'm trying to tell you is that LDL receptors can be recycled. The LDL is broken down, but the LDL receptor is not. It's recycled and it's put onto the surface again. I'm going to quiz you on this in the next slide. Okay, so I told you that inside of the cell, there was a specific kind of protein and it's responsible for creating the shape of that pit. What was the name of that protein? Good, somebody said, yeah, people are saying clathrin. Somebody asked a really good question. What will happen if there's no lysosome? There are very rare conditions where that happens, and it just leads to large accumulations. It's very rare. It's one of those really rare inborn disorders, and it just leads to accumulation of, eye cell disease, I think. And it just leads to accumulation of like everything inside the cells, and I don't think it's very compatible with life. But yeah, you can't break down any of the proteins or anything that's entering into your cells, so you pretty much can't function. But that's a good question. Okay, clathrin was the first one. Okay, and then I told you that we form like this endosome, but, oh, did I already give away the answer? Oh, I think I just told you the first answer of this. Okay, I'm not sure, but even if, okay, fine, we form our endosome, what was the cellular structure? What was the cellular structure that breaks down what was the organelle that breaks down the lipoprotein? Lysosomes break down, that's their role. The organelle that breaks down the lipoprotein is, yeah, okay. So the first one is endosome, I gave that to you, you're welcome. The other thing I want to tell you is something I really emphasized on. The lipoprotein is degraded, but the receptor is recycled. Yes, this is going to become really important because I'm going to tell you about statins and stuff, and that's why I'm really emphasizing this concept. Okay, you guys already answered this. Lysosomes are degraded. Okay, so you guys have to know this by now. LDL, does it deliver triglycerides or cholesterol? Cholesterol, right? This is easy. You guys know this by now, hopefully. But that's a really important concept, especially when I get to the disorders. Okay, good. So now I'm going to tell you why we hate LDL, like why we fear LDL so much, why we don't like cholesterol, okay? So I told you LDL is pretty much exclusively made out of cholesterol, right? It's not so much its effect on the cells, as much as it is the effect on our blood vessels, okay? So pretty much what happens is that I have my LDL. This is a normal LDL, nothing's happened to it. I told you there's only one apolipoprotein attached to LDL, it's ApoB100. The problem is that the LDL can undergo oxidation, right? Like just like everything in our body, for whatever reason, something else is going to steal an electron from it. And so the LDL can become oxidized, what we call oxidized LDL, okay? Specifically, I believe it's the apolipoprotein that gets oxidized. And I drew sort of this like yellow thing around it to make you think that like the oxidized LDL is the more unstable version. Okay, we don't like oxidized LDL. It's very unstable. And so what will happen is that this normal macrophage, okay, it has this receptor over here, okay? This receptor is called scavenger receptor class A. I need you guys to remember it's scavenger receptor class A, class A. I have a strong feeling this is going to be tested on. And class A, the reason you're going to remember class A is because I'm talking about atherosclerosis. So scavenger receptor class A is going to lead to atherosclerosis. So the A stands for atherosclerosis, okay? So my macrophage has this receptor called scavenger receptor class A, and the scavenger receptor class A is going to bind to only my oxidized LDL. The scavenger receptor doesn't do anything to my normal LDL, it doesn't care about this. The scavenger receptor class A only binds to my oxidized LDL, okay? And so once the scavenger receptor binds to my oxidized LDL, the macrophage is going to take in all of this fat, right, inside. Imagine there's a bunch of LDLs that are accumulating over here, and it forms what we call a foam cell. So this macrophage, once it intakes all this fat, all this LDL fat, it's going to form what we call a foam cell, okay? And the problem with foam cells is that we don't have a regulatory mechanism to make them stop. Like they will just keep accumulating in our body. Yeah, there's no feedback inhibition or anything like that. And the problem is that when the foam cells start to accumulate, they obviously have so much fat inside of them. So I have these foam cells that are accumulating, which is eventually going to form what we call an atherosclerotic plaque. What exactly is an oxidized LDL? It's literally just an LDL molecule that's lost an electron or lost a few electrons. Does that make sense? It's probably the apolipoprotein B that's lost the electrons. And so now that it's lost the electrons, it's less stable. Does that make sense? Okay. So I'm going to show this again in the next diagram, and I'm also going to quiz you guys on the terminology that I've taught you in this slide. Okay, so I really emphasized on this specific receptor that macrophages possess. What is the name of that receptor that recognizes the oxidized LDL? Yes, scavenger receptor class A. Class A because it leads to atherosclerosis, okay? Do not write class B, I will be very sad, okay? So yes, class A. Okay, so I told you that the scavenger receptor class A, they only recognized a certain form, a certain version of LDL. What version of LDL am I talking about? Okay, I see one answer. Can I get some more answers? Good. Oxidized LDL. Oxidized scavenger receptor A only recognizes oxidized LDL. And I told you that there's no regulatory mechanism. So no matter how much cholesterol I have, scavenger receptor A is just going to keep adding. I'm going to get this accumulation of foam cells, right? Oh, I spoiled the next one. Okay, the macrophages, once they take in the, once the macrophages, they take in oxidized LDL, what did they form? What is the name of the structure? Foam cells. Yes, good. Okay, and what is the name of the kind of plaque that forms on our blood vessel once these foam cells start to accumulate? What is the name of that plaque? Yes, atherosclerotic plaque. And then I told you that there's one specific lipoprotein that when you think of atherosclerosis, you have to think of this lipoprotein, right? When you think of cholesterol, you think of the bad parts of cholesterol, you think of atherosclerosis, what am I thinking of? What lipoprotein am I thinking of? Yes, LDL or LDLC, yes. Bad cholesterol, exactly. Okay, so hopefully that makes sense. You guys got this. Is a pretty complex topic, but you guys are doing pretty well. Okay, so I don't want you guys to get confused about this. Yes, HDL is made by the liver. Yeah, like that's totally true. But I told you that the role of HDL is not to deliver fat from the liver to the periphery. The role of HDL is to deliver the fat from the periphery back to the liver. Okay, so yes, it is originally made by the liver. This is true, but it's not delivering the fat from the liver to the periphery. It's doing the opposite. Okay. And then what kind of apolipoprotein did I tell you that is only found on HDL? Okay, I see one answer. Do we want some more answers? What apo? No, it's not apo. I think 100, you're saying ApoB100. Talking about HDL. ApoB100 is on VLDL, IDL, and LDL. Only those three. Only those ones. Yes, it's ApoA. ApoA, the capital A. It's going to come up later. So I know this is really...
The last school thing. Yeah.
I think someone's mic is on. But make sure that you guys differentiate that it's the apolipoprotein capital A. I know that's so annoying, but for some reason there is actually a difference between apolipoprotein capital A and apolipoprotein lowercase a. I'm going to be going over that towards the end of this presentation. So keep in mind that the apolipoprotein capital A is the one we find on HDL. So that's the answer to the first one. Yeah, apolipoprotein capital A1. And then I told you in the previous lecture, and I know I only mentioned this briefly, but if anybody can remember, there were two apolipoproteins or two classes of apolipoproteins that I told you HDL can donate to stuff like VLDL, chylomicrons, just temporarily. Yes, ApoE and ApoC. ApoE and ApoC2, exactly. So these are the more temporary ones, okay? And they just, the HDL sort of like holds them so that when they're needed, it will give the apolipoproteins to the VLDL or the chylomicrons. Okay, if you remember in my mnemonic, I said that yes, Hadil, HDL, she always gets A's. And then I told you that she also was the only one that got into this law college, admissions test. She was the only one that passed it. That was to remind you of this enzyme. What's the name of the enzyme? LCAT. Only interacts with HCl, okay? LCAT only interacts with HCl. And if you forget that, remember the function of LCAT. It's periphery to liver. Only one lipoprotein does periphery to liver. So yes, LCAT is the enzyme. It's activated by ApoA1. So the same way that we have ApoC2 activating LPL, the same mechanism is causing ApoA1 to activate LCAT, right? And I explained the mechanism of what LCAT does. Pretty much it's going to make the cholesterol into a cholesterol ester. And the cholesterol ester is going to be added to the HDL. Okay, so the HDL can't take cholesterol on its own. HDL needs the cholesterol to be converted to a cholesterol ester. The LCAT is responsible for that. So the LCAT converts cholesterol to cholesterol ester by adding a fatty acid. And then the cholesterol ester will be transported inside of the HDL. And the HDL transports cholesterol from where to where? I've said this like 10 times, so I know you guys know this. From where to where? Yeah, okay, I have one answer. Can I get some more answers? Because I want to make sure everyone knows this. Yes, from my peripheral tissue to my liver. When I say peripheral tissue, I literally mean anywhere but my liver, you know? Peripheral tissue to my liver, exactly. And then I told you it's called reverse cholesterol transport. That's why we really like this. Yeah, see, this is what makes HDL the good cholesterol. And then just as sort of a side note, especially once you get into third year CVP, you guys are going to notice, and I'm sure you guys are kind of aware of this, that cardiovascular diseases, especially stuff like myocardial infarction, hypercholesterolemia, disproportionately affect men. Okay, so men are way more likely to have something like a heart attack. And the reason for that is because estrogen is sort of protective against that. Similarly, postmenopausal women, so women who no longer produce estrogen because they've like gotten older, they are also at an increased risk because estrogen raises HDL levels, and HDL is the good cholesterol. Okay, so they have less of that protective mechanism. Okay, and then obviously exercise increases HDL because exercise is good, and so it's going to increase the good sort of lipoprotein. Okay, so I talked to you in this vague sense about why HDL is good, right? And I really emphasized on the peripheral to the liver, peripheral to the liver. I would argue the second mechanism is even more important, especially when I'm talking about atherosclerosis. Yes, it's important that I don't want lipids or cholesterol accumulating in my peripheral tissue, but I especially do not want cholesterol inside of my blood vessel. And we talked about why. We talked about if I have too much cholesterol accumulating in my blood vessel, it's going to lead to a plaque. And if I have hardening of my arteries, there's going to be less blood flow. And just imagine, if I have less blood flow going into my heart, I'm going to get a heart attack. If I have less blood flow going into my brain, I'm going to get a stroke. So stuff like that. So we really don't want cholesterol just accumulating in our blood vessels, right? So I also emphasize a lot on how I want you to remember scavenger receptor A for LDL, because I do not want you to mix up scavenger receptor B1. Scavenger receptor B1 is responsible for taking the HDL, it's responsible for taking the HDL back into the liver. You see over here, the receptors over here sticking out of the liver, sticking out of the hepatocyte. And so when the HDL comes, it's going to bind using its ApoE. And so then the HDL can be taken into the liver. Okay, so that's the function of scavenger receptor B1. It's so that hepatocytes can take in HDL. So different from scavenger receptor A. So different from scavenger receptor A, right? So I really want you guys to differentiate between the two. I also told you that one of the functions, I told you that HDL carries cholesterol mostly, and I told you that cholesterol is used to make steroid hormones. So it shouldn't surprise you that one of the roles of HDL is not only to go to the hepatocytes, it's also to go to tissues that need to make steroid hormones. Okay, so I think somebody's asking the difference between scavenger receptor B1 and scavenger receptor A. Okay, so scavenger receptor B1, it's located on a hepatocyte, okay? And pretty much what happens is that when my HDL comes, my HDL molecule is going to bind to the scavenger receptor B1 on my hepatocyte, and this is going to induce endocytosis. It's going to make the HDL enter into the hepatocyte, okay? So scavenger receptor B1 makes the HDL enter into my liver, enter into my hepatocyte. Okay, so it literally just makes the HDL enter in just for storage into the liver. Versus, I told you scavenger receptor A1 is the evil one. We don't like this one. Scavenger receptor, not A1, sorry. Scavenger receptor A is the one that's located, let me show you. Scavenger receptor A, I told you, is the one that's located on my macrophage, and it recognizes oxidized LDL. And once it recognizes oxidized LDL, we get foam cells. And once we get an accumulation of foam cells, we get atherosclerotic plaque. Okay, so these two things are completely different mechanisms. Scavenger receptor A, think of atherosclerosis, think of oxidized LDL, think of macrophage. It's located on a macrophage. But then scavenger receptor B1, think of liver and think of HDL. Okay. Okay, so if you guys remember, so over here, as you can see, HDL, the cholesterol, can be transferred onto other like lipoproteins. We talked about VLDL, we talked about like chylomicrons. It can actually also be transported, it can also be transported to IDL. So the HDL cholesterol or the cholesterol ester that's inside of the HDL can be transferred into other lipoproteins, such as VLDL, chylomicrons, IDL. I told you that there was a protein, a very specific protein that is responsible for this. What is the name of that protein that transfers the cholesterol from HDL to other lipoproteins? Okay, I see one answer. Does anybody else want to answer? Okay, I don't think there's any other answers, but that's correct. CETP. Yeah. CETP, you can just guess by the name, cholesterol ester transfer protein. It's responsible for transferring the cholesterol that's inside HDL into other lipoproteins, such as IDL, VLDL, or chylomicrons. And I think somebody asked in the, why does it, okay, I don't know why it does this. Like, I don't know, like I can't rationalize why, but I think somebody asked in the previous one why this would be bad. And the answer to that is like, or like, yeah, like why is it bad if the cholesterol ends up in the VLDL, right? Or like why, I think the way that they phrase it is that it's like, why is it damaging the VLDL? It's not that it's damaging the VLDL. The VLDL isn't being damaged. It's that we're going to get more of the cholesterol that's in our body is going to be stored in the VLDL, or it's more of the cholesterol is going to be stored in IDL, or more of the cholesterol, yeah, like for example, for those two. And the problem with that is that if you remember, I told you that VLDL and IDL eventually turn into LDL. And so ultimately, if my HDL is transferring its cholesterol into VLDL or IDL, it's going that cholesterol that was originally in HDL is now going to end up in LDL. And we don't like that. LDL is the bad cholesterol. I don't know why this protein exists or anything like that. I would assume that, you know, maybe, I don't know, it would be good if we were starving or like in the earlier days or whatever, when we didn't have access to food, we wanted like more cholesterol to end up in LDL. But nowadays we don't need that much cholesterol. And so something like that. But the point is that like we don't like CETP because CETP moves the cholesterol from HDL, which is the good guy, to the bad guy, which is like VLDL, IDL. Does that make sense? Yeah? Okay, hopefully. I know it's a little bit confusing. But that's why you can kind of think of CETP as the bad guy, because it's moving cholesterol from a place that we like it to a place that we don't like it. And then this should make sense to you guys now. So if I inhibit CETP, that means that more of the cholesterol is going to be staying in my HDL. So sort of like the circulating HDL levels. Like my HDL won't be broken down as much, pretty much, because the cholesterol will be forced to stay inside of my HDL. So I believe what Dr. Ahmed told us is that you were actually looking for drugs that would inhibit CETP because we want to raise HDL levels because we like HDL. HDL is the hero, it's the angel. So CETP is bad, yeah, I would say, yeah, pretty much. So we would like drugs nowadays that would inhibit it. Okay, so this was actually in the first PowerPoint, but I moved it over here because I think it's more relevant in terms of cholesterol. I think the main idea that they're trying to tell you is that like the reason we're so afraid of increased cholesterol levels is because it stays so long in our blood. Like if you look at glucose, we're still afraid of increased glucose levels. So don't get me wrong, like diabetes is a problem. But at the very least, we have enough mechanisms in our body that like glucose can be cleared from the blood. In comparison, the mechanisms that remove cholesterol from our blood just don't act as fast. And so the cholesterol is going to stay longer in our blood, right? Like the peak is somewhere around 6 versus glucose is like less than 3. It's already cleared by 3 hours. You know what I mean? And why we're so scared of that is that cholesterol that stays in our blood, you know, is going to eventually form an atherosclerotic plaque. So I think that's just the idea they're trying to convey over here. Okay, so this is to quiz you guys. So I told you that HDL, sorry, let me just remember. Okay, yeah, I told you there was an enzyme that only interacts with HDL. What was that enzyme? The role of that enzyme was to take the cholesterol that's in my peripheral tissue, bring it back to my liver. What was the name of that enzyme? Yeah, okay, I see one answer. Does anybody else want to answer? The enzyme, not the receptor. SRB1 is the receptor that's located on my liver, but I'm talking about the enzyme. Yes, LCAT is the enzyme. Okay, and then what is the apolipoprotein that I need to activate LCAT? What is the name of the apolipoprotein that's located on HDL that I need to activate LCAT? Good. Oh, somebody said ApoC2. No, ApoC2 is needed to activate LPL. It's needed to activate lipoprotein lipase, which acts on VLDL and chylomicrons, because LPL breaks down triglycerides. Okay, but I'm not talking about triglycerides over here. I'm talking about the cholesterol that's inside of HDL. Okay, and that enzyme is different. It's not LPL anymore. It's LCAT. LCAT is the enzyme, yes. If you're struggling to remember which one's the receptor, which one's the enzyme, I would recommend learning the full name. So I believe it's like lecithin cholesterol isol transferase, and anything that ends with ase is always going to be an enzyme. So LCAT is the enzyme that breaks down, or doesn't break down the cholesterol, it converts the cholesterol so that it can be added to the HDL. Okay, yeah, so somebody's saying we're going over the apolipoproteins at the end. For sure, I will make sure to go over that. So yeah, I told you that HDL, it has apolipoprotein A, and that activates LCAT. Okay, and then I told you that macrophages, they have something, somebody asking, what is the other one? What is the other what? Oh, ApoA1. Okay, yeah, sorry. So ApoA1 is the lipoprotein that's, remember A, like Hadil only gets A's, like she's the only one that gets A's, because HDL is the only one that has apolipoprotein A, capital A. And apolipoprotein A1 is what activates the LCAT enzyme, law college admissions test. She's the only one, Hadil, HDL is the only one that passed the LCAT, because HDL is the only one that interacts with this enzyme. And I told you what the function of the enzyme was, which is to convert the cholesterol into cholesterol ester, so that now the cholesterol that's in my peripheral tissues or in my blood vessels can go to my liver through HDL, okay? And then in the second part of that, a completely different mechanism, I'm shifting gears, I told you that the problem with having high cholesterol in our blood is that I have macrophages. The macrophages have these very specific receptors called scavenger receptor A, scavenger receptor A, not B1, scavenger receptor A, and that the scavenger receptor A will phagocytose what kind of lipoprotein? Yes, oxidized, specifically oxidized LDL. Oxidized LDL, good. And that leads to a plaque formation. Plaques are bad in our blood vessels because they're going to decrease blood flow, leading to either heart attack or stroke or long list of complications. Okay, this is good because we will review some of the apolipoproteins now, and I'll make sure to go over them again at the end. But let's just review, for example, chylomicrons. There's a very specific apolipoprotein I told you is only found on chylomicrons. What apolipoprotein is only found on chylomicrons? I gave you a mnemonic of like triglycerides is for E, which should remind you of the diet, E for eat. And then you remember eat, you should also remember the past tense of eat, which is ate. If you remember 8, you should remember ApoB48. Yes, okay, good. So ApoB48 is only found in chylomicrons. Just remember this, chylomicrons, diet. Think of food, think of eating food, you know. Somebody said ApoB100. No, it's not ApoB100. ApoB100 is only VLDL, IDL, LDL. Okay, just those three. Those ones that are coming like straight from the liver. And the mnemonic I have for that is like be 100% prepared for your delivery, right? So it's like the mother liver is telling them, it's giving them ApoB100, so it's telling them to be 100% prepared so they can deliver their fats or their lipids. Okay, yeah, so you got that right. Oh, I did this in the wrong order. Oh man, okay, so you guys know now that LDL, the only one that's present on it is B100. And then the last thing I want to test on the slide, although I definitely will go over it in the end, is that I told you that there was a receptor that is responsible for uptaking HDL inside of the liver, inside the hepatocytes. What is that? Okay, I see one answer. Can I get some more answers? What is the name of the receptor that's on hepatocytes that takes in HDL? Okay, yeah, 2 people said scavenger B1. Yes, scavenger B1. Okay, so if you remember in the last diagram, I made you guys, we stopped at the part where I take in the LDL, I break it down, blah, 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 and then I end up with free cholesterol. but I didn't tell you what happens to that free cholesterol. And there's three main things that I want you to know about what happens, the effects kind of the cholesterol inside of a cell. Okay, so let me just make sure I'm getting all of these. Okay, yeah, okay, good. So once I have cholesterol that enters into my cell, this is going to lead to a high cholesterol concentration inside of my cell, right? So I'm talking about a high cholesterol concentration. I believe, honestly, we're specifically talking about the hepatocyte, so I'm just going to refer this as a hepatocyte. Let's say the cholesterol enters into my hepatocyte, and I have a high concentration of cholesterol inside of my hepatocyte, okay? This is going to lead to three things, and just think about this logically, okay? So one, I need to store my cholesterol, but I told you that cholesterol is really not soluble, and so we need to convert it into a cholesterol ester. We need to add a fatty acid on top of it in order for it to be stored. And this is through an enzyme that's called ACAT, not LCAT, okay, not the law admissions test. No. That's completely different. I try to create a mnemonic to separate the two, but this is ACAT, okay? ACAT is responsible for making the cholesterol into a cholesterol ester for storage, okay, inside of a cell. That's the first thing that's going to happen. Okay, the second thing that's going to happen is that if you think about this again, cholesterol just entered into my cell. There's a high concentration of cholesterol. Do you think my cell wants to make more cholesterol, any more cholesterol than it already has? There's already such a high concentration inside of the cell. I don't want to have too much, right? I don't want to keep making, I don't want to keep making any more cholesterol. No, I don't. So what's going to happen is that my cholesterol is going to signal to this very specific enzyme called HMG-CoA reductase, okay? And the function of HMG-CoA reductase is to make cholesterol. That's all you need to know about it, is that it makes cholesterol, okay? HMG-CoA reductase, ACE, it's an enzyme. So it's an enzyme involved in the pathway of making cholesterol. So what's going to happen is if I already have such a high concentration of cholesterol, my cholesterol is going to tell HMG-CoA reductase to stop. It's going to inhibit HMG-CoA reductase, so the cholesterol synthesis pathway stops, and I don't make any more cholesterol because I already have enough cholesterol, okay? That's the second thing that's happening. The third thing that's happening, and this is really, really, really important, I'm going to decrease the expression, right? Like I'm going to decrease the DNA expression of LDL receptors. This sounds a little bit confusing, but just think it through. Okay, I already have a high concentration of cholesterol inside of my cell. If I keep making LDL receptors that are going onto the surface of my cell, and the LDL receptors keep bringing in more LDL, more cholesterol, more cholesterol, more cholesterol, this is bad. I don't want any more LDL receptors because I already have a high concentration of cholesterol inside of my cell. So what's going to happen is that the high concentration of cholesterol inside of my cell is going to inhibit the synthesis of LDL receptors. So I have less LDL receptors being made. Less LDL receptors means less LDL entering into my cell. Less LDL entering into my cell means less cholesterol in my cell. And that's what we want, okay? Because I already started off with a high concentration of cholesterol. So it's pretty much feedback inhibition. So the three things again is storage. What I want you to remember about the storage is that it involves the ACAT enzyme, okay? The second thing I want you to remember is that we are stopping the synthesis of cholesterol by inhibiting the HMG-CoA reductase enzyme. And the third thing I want you to remember is that we are stopping the formation of LDL receptors, literally at the DNA level. We're stopping the synthesis of LDL receptors because we don't want any more LDL receptors because it would just lead to more and more cholesterol, and we already have enough cholesterol, okay? So hopefully those three make sense. I'm going to test you on it in the next level and the next slide. Make sure you guys pay attention to the small details as well. So for example, over here, I told you that cholesterol decreases the synthesis of LDL receptors. sometimes what they'll do on an exam to make it a little tricky is that they'll have another option that says degrades the LDL receptors, for example. You know what I mean? Like the cholesterol, the high amount of cholesterol will degrade the LDL receptor. But that is not what I said. That's not what I said. I didn't say the cholesterol degrades these receptors. No, I said it's going to go to the DNA. It's going to say, hey, stop making LDL receptors. So it's going to stop the synthesis of LDL receptors. So I want you guys to be able to differentiate between those two things. I'm not breaking them down. I'm just stopping them from being formed. Those kind of details kind of matter. Okay, so I told you that there were 3 effects of cholesterol once it enters into the cell. One is that it activates this enzyme so that it can be more soluble to store into the cell. What is the name of that enzyme? Okay, I see one answer. Yes. Okay, good. Yeah, ACAT. ACAT is the name of the enzyme. This is the full name over here. It ends with an ACE because it's an enzyme. I told you that we wanted to stop the expression of this gene, which ultimately led to this protein or this enzyme that was involved in cholesterol synthesis. What is the name of the enzyme that is involved in cholesterol synthesis? Yes, HMG-CoA reductase. Again, notice over here, it's stopping it at the level of the gene, okay? Stopping, and I'm preventing the synthesis. Okay, I'm not degrading these, I'm preventing the synthesis. Okay, I also told you that there was another thing that it also represses. Yes, inhibits production of LDL receptors. Good, at the gene level. Good. Okay, and then so all of the stuff that I just talked about over here, all the stuff, especially over here when I'm talking about at the gene level. The thing is, I need some specific protein that's called a sterile, what's it called? Sterile receptor? Yeah, sterile response element binding protein 2, SREBP2. This is a protein, and this protein is called a transcription factor. And pretty much what they're trying to tell you is that like this transcription factor is needed in this process. It's needed to suppress the gene expression of HMG-CoA reductase. It's needed to suppress the gene expression in LDL receptors. Or I should phrase that better, actually. Let me try that again. It's more so that we need to inhibit SREBP2. So we need to inhibit SREBP2 in order to stop the synthesis, to stop the synthesis of HMG-CoA reductase and stop the synthesis of LDL receptors. So normally, SREBP2, normally, just think about this, like what is the normal function of this transcription factor? It's going to promote the expression of LDL receptors. Okay, normally SREBP2, this transcription factor, promotes the expression of LDL receptors. So what I need to do when I have a high amount of cholesterol is the high amount of cholesterol needs to go to SREBP2, needs to go to the specific transcription factor and inhibit it, so that if I inhibit SREBP2, I will inhibit HMG-CoA reductase expression, and I will inhibit LDL receptor expression. So the mnemonic that I want you guys to remember for SREBP2 is I'm going to pretend that it stands for something else. I'm going to pretend that it stands for statins reduce everyone's blood pressure, okay? Can you repeat its function? So normally, okay, SRBP2, BEP2, once it is activated, it is going to lead to, for example, expression of LDL receptors, leading to more LDL receptors that are going to be present on the cell surface, normally. But when I have a high amount of cholesterol, the high amount of cholesterol is going to inhibit SREBP2. And if I inhibit SREBP2, then HMG-CoA reductase expression will be reduced and LDL receptor expression will be reduced. Okay, so do we understand like the normal function? Okay, so somebody asked, and this is a good question, but if it's not taken in by the cell because low LDL receptor, wouldn't that increase blood cholesterol in the blood, which is much worse? That's a really good question. And you've actually already explained the mechanism of statins. That's what we do. That's exactly what we do. And so I'm going to explain that in statins, but you're right. When we have less LDL receptor on the surface of the cell, that means that more cholesterol is staying in the blood, and that's kind of bad. But it's only bad if it's in excess. If everything stays in homeostasis, it's fine. Okay, so I'm gonna explain statins at the end, but pretty much what somebody asked is that, like, I just want you guys to understand the relationship between, like, if I have LDL receptors, the more LDL receptors I have, the more LDL is being taken in, and so it lowers blood cholesterol. I just want you to understand that relationship right now. I'll explain the statin stuff later. Okay, so I just want you to understand this relationship. LDL receptors, more LDL is being taken in. The more LDL is being taken in, the cholesterol is being removed from the blood. This part is the blood, this part is the cell. So if I have more receptors, more LDL is entering into the cell, and that means that more cholesterol is in the cell, and therefore less cholesterol is in the blood. I just want you to understand that relationship for now. Does that make sense? Yes? Okay, good. And then I'll explain the other part later. Okay, yeah. And I'll explain this mnemonic later as well, EBP2. For me, it's really hard to memorize stuff like this, so I always come up with mnemonics. And it's a little bit of a steam. Doesn't that contradict? I'm not sure what you're asking. Doesn't what contradicts what we said about less receptors? Oh, maybe you're talking about my mnemonic. Don't think about my mnemonic right now, actually, then. It'll make more sense when I get to statins then. Okay, so yeah, this is just the SREBP2. And as you can see, it acts like on a gene level. And I told you that, like, the SREBP2 is needed because the more once we activate SREBP2, it's going to lead to more expression of HMG-CoA reductase and LDL receptors. Okay, so now I'm going to go into the disorders, and then after the disorders, I promise I'll go into the statins. Okay, so what I want you guys to know and how I want you guys to remember this is that this is called a type 1 hyperlipoproteinemia. To remember that the type 1 is hyperkylomicronemia, what I want you to pretend is that the 1 is actually a lowercase l. And because there's only one lowercase l inside of chylomicron, that can help you remember that, like, okay, there's only one lowercase l over here, then that means that this is the hyperkylomicronemia. Okay, kylo, there's one lowercase l here, and it looks similar to the type 1, right? Like these two symbols look similar. Okay, so you guys have to know this. Chylomicrons, what kind of fats do they carry? Mostly. Is it triglycerides or cholesterol? Yes, triglycerides. So obviously, if I have high amounts of chylomicrons, it's going to lead to high amounts of triglycerides. Okay. So if you guys remember, I said that there was this enzyme that only acts on VLDL and chylomicrons because its role was to break down the triglyceride. Actually, its role was to cleave the triglyceride into two parts, okay? And it only acted, yeah, on VLDL and chylomicrons. What is the name of the enzyme and what is the name of the cofactor? Tell me both. Okay, ApoC2, good. ApoC2. And what is the name of the enzyme? Okay, somebody said LPL, so yeah. So obviously what happens is that if I have a deficiency in LPL, or if I have a deficiency in ApoC2, I can no longer break down my chylomicrons, and that's why they're accumulating. Okay, but if you guys remember in the first presentation, I told you that usually if a patient is going to present to you with high amounts of triglycerides, you should be thinking more in the sense of VLDL, and we'll go into that disorder next, or actually last, because I just want you guys to be aware of the fact that hyperkylomicronemia is more rare compared to high VLDL. So both of these conditions are similar because they're both going to lead to high amounts of triglycerides, but high chylomicronemia is just more rare. Okay, so that's why if somebody comes with high triglycerides, we assume it's VLDL. Okay. And then what I really, really want you guys to also know, okay, so obviously these clinical manifestations are important and you will have to memorize them. I want you guys to be able to remember that the high amount of triglycerides or hyperkylomicronemia is going to lead to xanthomas that are on the skin, not on the tendons. If it's high chylomicrons, it's going to lead to these little xanthomas. Xanthomas are just like bumps on your skin. Like that's all you have to know at this level. So he's like usually like maybe itchy, but like these bumps on your skin called cutaneous. Cutaneous means skin. And the xanthoma I told you is the bump. And that's specific, or it indicates high amounts of chylomicrons. Okay. So you have to be able to differentiate, are these bumps on the skin or are they on the tendons? Because in the next slide, I'm going to show you that these bumps can also be on your tendons, not on your skin. So if it's on your skin, you should be thinking of chylomicrons, too much chylomicrons. Pretty much the triglycerides are depositing and that's what's happening, like in your skin. The other thing, and I would argue this one's more important, and I really, really want you guys to remember this. I told you in my mnemonic, triglycerides, E, it looks like the letter E. E is for eating. Your pancreas is an organ that is involved in eating, okay? So that's why I want you guys to remember that if you see something like pancreatitis, that's a good indication of too high triglycerides. And pretty much what's happening is that your pancreas is responsible for breaking down triglycerides, okay? So when I have too much triglycerides that are in my blood, the pancreas is working too much and too hard, and that's eventually going to lead to inflammation, okay? But the main thing just with the mnemonic is the triglycerides look like E. You should think of eating, you should think of the dietary tract, the GI tract, and so you should think of the pancreas, not atherosclerosis. I've never mentioned triglycerides anywhere in atherosclerosis. I only said cholesterol. I said LDL, and I said when you think of LDL, you have to be thinking of cholesterol. And in my mnemonic, I showed you that like there's the cholesterol and there was the heart around the people that, you know, the guy that was proposing. And that should remind you atherosclerosis is associated with cholesterol. Atherosclerosis is not associated with triglycerides. So you should be able to reason out in your head right now, even if the only thing that you knew was that, okay, this is high amounts of chylomicrons. If I have high amount of chylomicrons, that means high amount of triglycerides. Triglycerides have nothing to do with atherosclerosis, have nothing to do with atherosclerosis. Atherosclerosis is only cholesterol. Instead, my triglycerides are associated with my GI tract. And so if I have too much of them, my pancreas is going to have to work overtime, eventually going to lead to inflammation. It's going to lead to pancreatitis. I also do want you to remember the cutaneous xanthomas though. Cutaneous xanthomas is associated with hyperkylomicronemia. Okay, so remember I told you, you need to be able to differentiate in a clinical scenario, is the bump on the skin or is it a tendon? Is it on the tendon? This is important because it presents in different syndromes, right, or different disorders. So for example, I told you in hyperkylomicronemia, the xanthomas are going to be on the skin. The bumps are going to be on the skin. Versus over here in type 2, the xanthomas are on the tendon. Look at that, it's on the tendon. But let's talk about what type 2 hyperlipoproteinemia is. So I said in the previous mnemonic or whatever, that the way that you're going to remember this is that the type 1 looks like a lowercase l, and there's only one L in chylomicron. Versus in type 2, I see two L's, right? I know they're actually like Roman numerals, but let's pretend they're L's. And LDL has two L's. Okay, so type 2, two L's. And that means that it's elevated LDL. So that's a way that you can memorize it, hopefully. If I have too much LDL, which type of fat is going to be too high? Am I going to have too high triglycerides, or am I going to have too high cholesterol? You guys tell me. Cholesterol, right? It has to, you have to say this. LDL, C, LDL, you have to be thinking too high cholesterol, okay? And I told you what I wanted you to know about this is that the type 2 hyperlipoproteinemia is caused by too high LDL levels. Too high LDL levels means too high cholesterol levels. One of the clinical manifestations is tendon xanthomas, not cutaneous xanthomas. And then if you just think about this logically, remember how I told you on the liver, there was this receptor that's called the LDL receptor? Imagine if I just don't have that receptor. That means I can't take in the LDL. If I can't take in the LDL into my liver, the LDL is forced to stay in my blood. If the LDL is forced to stay in my blood, the cholesterol is staying in my blood. So that's how LDL receptor deficiency is leading to hypercholesterolemia. And he's just differentiating between type 1 and type 2A and type 2B. Type 2A is like you don't have the receptor at all. You know, like it's like a genetic problem. Like you're just not expressing that receptor, versus type 2B is like a lifestyle thing. So it's not about the receptor, it's just that they're eating or, you know, whatever like risk factors or obesity is leading to too high cholesterol in the blood. And so that they get hypercholesterolemia. Like it's more straightforward versus the type 2A. I don't think you guys need to know this, but the type 2A is the one that has the receptor deficiency. That's what I'm trying to say. Okay, and I told you that when you think of LDL and you think of cholesterol, there's one major clinical side effect, something we're so afraid of. What are we afraid of? What are we afraid that this is going to lead to? I see one answer. What can too high cholesterol in my blood lead to? Good, atherosclerosis. Okay, so I hope you guys don't get this wrong in the exam. The previous one, it's not going to lead to atherosclerosis. It's high triglycerides. Triglycerides I never mentioned with atherosclerosis. But I told you LDL and cholesterol is associated with atherosclerosis. So you should be able to differentiate that. Okay, and then the third one we're going to talk about is actually type 4. The V in the 4 should remind you that this is elevated VLDL, right? The VLDL, okay? Elevated VLDL is going to lead, so I told you the VLDL has both triglycerides and cholesterol, but it has more of one type of lipid. What is that type of lipid that I'm talking about? What lipid is mostly in VLDL? It's like 60% of VLDL is this one type of lipid. Is it triglyceride or is it? Yes, it's triglyceride. So it should make sense to you that, again, if you remember this and you start from the beginning, okay, they present you a scenario. It has type 4. I look at the 4. I look at the V. Once I see the V, I remember, okay, the V stands for VLDL. You don't have to memorize that it leads to high triglycerides. You already know it leads to high triglycerides because you already know that VLDL is mostly made of triglyceride. Okay, yeah, triglycerides is elevated more than the extent of cholesterol. Again, unfortunately, you guys have chosen medicine, and so that means you have to think in shades of gray. Okay, so that means that yes, I did tell you triglyceride is more elevated. That does not mean that my cholesterol is not going. It does have some cholesterol, exactly. The VLDL still has some cholesterol, and it's still significant. Remember when I was doing my calculation? Remember when I tried to calculate my cholesterol? I said there were 3 that you need to calculate with. I said LDL, I said HDL, and I still said VLDL. You still have to calculate the cholesterol that's in VLDL, even if it's not, even if there still is more triglycerides, okay? So I need you guys to differentiate between the fact that like VLDL still has enough cholesterol that it can still lead to atherosclerosis, okay? Versus my chylomicrons, the cholesterol is so little that it still won't lead to atherosclerosis, okay? Does that make sense? Like my type 4 VLDL, it will lead to high triglycerides more. It'll lead to very high levels of triglycerides, but it still will increase my cholesterol level. Does the differences between these 3 make sense? Like why type 4 leads to atherosclerosis, why type 1 doesn't? Yes? Because these disorders are pretty important, so I want everyone to be able to differentiate. Okay. People are saying yes. Okay. Oh, this was something else that I really want to mention. For whatever reason, biochemists have decided that hypertriglyceridemia is going to specifically reference VLDL. Yes, I know. Hypertriglyceridemia, high levels of triglyceride in the blood, could also be caused by high levels of chylomicrons. But the biochemists decided that no, if I specifically say this phrase, hypertriglyceridemia, I'm specifically saying VLDL. If I want to say high levels of chylomicrons, I have to be more specific. I have to say specifically hyperkylomicronemia. Okay? Okay, so I kind of already gave the answer to this bottom one, but for the second one, hypercholesterolemia, what is it caused by? What kind of lipoprotein? Yes, LDL. An example of what could lead to that is if you don't have LDL receptors, right? You're not taking in the LDL. Good. Okay, so hypertriglyceridemia, think VLDL. Hyperkylomicronemia gives you the answer in it, chylomicrons, too many chylomicrons. And hopefully this middle part makes sense as well. You know, that like chylomicrons are 90% triglycerides, so triglycerides will increase at a very high rate. LDL is mostly cholesterol, so cholesterol will increase at a very high rate. VLDL is sort of a combination of both, but it's more triglycerides than it is cholesterol. Okay, so remember earlier I kind of mentioned to you that unfortunately there is a difference between apolipoprotein capital A and there is a difference between apolipoprotein lowercase a. I don't know why. I don't know why they decided to name it like this. It's very confusing. But apolipoprotein capital A, especially capital A1, was the one that I told you that was associated with HDL, apolipoprotein capital A. But apolipoprotein lowercase a is an entirely different thing. I'm going to explain what the difference is right now, or I'm going to explain what apolipoprotein lowercase a is. Okay, so over here, I have my normal LDL, right? It's mostly filled with cholesterol. It's the bad lipoprotein, and I told you it only has one apolipoprotein attached to it. And that apolipoprotein is ApoB100, okay? And what happens is that everybody in their body has this other form of LDL, okay, that looks like this instead. So it looks similarly, it has an LDL, I have my ApoB100, however, I have this like extra structure sticking off of my ApoB100, and this structure over here is called a disulfide bridge, and this extra structure is going to change the ApoB100 into an apolipoprotein lowercase a. So what I'm trying to tell you is that the ApoB100 normally kind of looks like this, but sometimes the LDL in our body is also going to look like this. Instead of an ApoB100, it's going to look like an apolipoprotein lowercase a, which is just this extra disulfide bridge attached to it, okay? And everybody has a combination of these two. Everybody in our body, we have this normal LDL, and we also have this version of LDL. The problem is the percentages. Ideally, even though LDL is the bad guy or whatever, we would still prefer this version of LDL, this normal version of LDL, instead of this version of LDL. And the reason for that is that this version of LDL, which I'm going to call lipoprotein lowercase a, so this entire structure over here, this entire LDL is referenced as lipoprotein lowercase a, okay? And the reason that this version of LDL is worse is because it's more likely to get oxidized. And I told you what happens when an LDL gets oxidized. Once an LDL gets oxidized, it can then be recognized by the scavenger receptor class A that's on my macrophage. And if my macrophage recognizes my oxidized LDL, it's going to phagocytose it. It's going to lead to foam cell formation. This is going to lead to atherosclerotic plaque. So the difference between these two is that lipoprotein lowercase a, which refers to this entire structure over here, is more likely to get oxidized. We do not want to oxidize LDL. So we oxidize LDL, it will eventually lead to higher risk of atherosclerosis, okay? So I know it's really annoying, but I just want to emphasize one more time. When I'm talking about the protein alone, I can say apolipoprotein lowercase a, but when you talk about the entire structure, this entire structure over here, this is called lipoprotein lowercase a. So this is lipoprotein lowercase a, and the small protein over here is called apolipoprotein lowercase a. So all I'm saying is that if I have an LDL and I attach apolipoprotein lowercase a to it, the LDL, I will now refer to it as lipoprotein lowercase a. Okay, and this is specific to LDL. As soon as you think lipoprotein lowercase a, you should be thinking LDL only. It doesn't happen to other lipoproteins, okay? So hopefully that makes sense. If it doesn't, I'm going to explain it again in the next slide. Okay, so this is just the diagram that was put on your lecture. So again, this entire structure over here is referenced as lipoprotein lowercase a. And it's because it has this extra structure attached, which is called apolipoprotein lowercase a. Okay, so the rest of the part of the LDL is totally normal. Okay, I have my LDL and I have my ApoB100. Okay, and the problem with apolipoprotein lowercase a, or my problem with lipoprotein lowercase a, that's more likely to get oxidized. And I don't want my LDL to get oxidized because then it can lead to atherosclerosis. Okay. Okay, so let's quiz you guys now. So I told you that lipoprotein lowercase a is a variant of LDL, LDL only. Okay, and it has an apolipoprotein lowercase a that is bonded to the apolipoprotein that's already present on my LDL. What is the name of that apolipoprotein that is already present on my LDL? There's only one apolipoprotein present on my LDL normally. There's only one. Okay, one person answered. Yeah, let's get some more answers, please. Good. ApoB100. Okay, so just showing the diagram again. Oops. This is my ApoB100. And attached to my ApoB100 is my apolipoprotein lowercase a. Okay, you guys are right. And I told you that apolipoprotein lowercase a, it varies between person to person. And depending on how much apolipoprotein lowercase a you have, it depends on your risk. It will increase. So the more apolipoprotein lowercase a that you have, the higher your risk of atherosclerosis is. Okay. I already told you the second answer again. I already spoiled it. Yep. What is it a risk for? The elevated lipoprotein lowercase a. What is it a risk for? Yeah, atherosclerosis. Hopefully you guys understand why. And it should make sense why it doesn't respond to dietary manipulations, because it's like, it's just the form of LDL that you happen to have in your body. You know what I mean? Like, it's not about how much cholesterol you have. Exactly. It's about what that structure of the LDL is. And there's nothing we can really do to change that. Not really. Okay, so I know this slide looks really dense. I think the main concept they want to make you guys understand is that there are two types of hyperlipidemias. I can either have a hyperlipidemia that is because I have too much triglyceride, or I can have a hyperlipidemia that is because I have too much cholesterol, right? And I think what they want you to understand is that if you have a hyperlipidemia that is because you have too much triglyceride, the management differs a little bit. You want to restrict their carbohydrate use. Okay, and the reason that's different is that when you have hyperlipidemia that's due to too much cholesterol, you want to limit their saturated fatty acid intake. I don't know why exactly. I don't know why it's saturated fatty acid for cholesterol and why it's carbohydrates for triglycerides. I couldn't find anything when I looked for it, but I think they just want you to understand that for people that have high levels of triglyceride, you want to limit their carbohydrates, and that people who have high levels of cholesterol, you want to limit their saturated fatty acid. I just want to remind you guys over here that saturated fatty acids are the bad ones, and polyunsaturated fats are the good ones. And the reason that is because I'm going to try to annotate. Let's see if I can. But like saturated fatty acids, they are linear. Okay, saturated fatty acids are linear, they're straight. So they look like this. If something is like this, is more straight, exactly, but my polyunsaturated fatty acids, they look like this. They have what we call kinks, okay? And they're bent. They have a lot more bent. Between the two of these, which of these looks more stackable? Which one do you think would aggregate more, would stack together more? Would it be the ones that are very straight, or would it be the ones that are all crooked? No, the first ones, the straight ones. The straight ones are so easy to stack. They fit so nicely on top of each other. Maybe it's because I drew this wrong, but like, if you could imagine trying to stack this, like you would have one going here, you'd have one going here. You know, they wouldn't really stack nicely. Versus the linear ones are so easy to stack on top of each other. They fit so nicely. So the linear ones, my saturated fatty acids, the problem is that because they're stacking, it's more easier for them to like clog my arteries. You know what I mean? They're going to aggregate together more easily. And actually, when you think about it, when you think of where you get, I lost my mouse. Where's my mouse? Oh my God. 1 second, guys, sorry. Ooh, okay, I don't know what's happening. Sorry, guys, 1 second. Okay, there. If you think about what my sources are for saturated fatty acids and my unsaturated ones, you guys will notice that my saturated fatty acids come from solid food. So for example, butter. Butter has saturated fatty acid, okay? And the reason that is that because because these saturated fatty acids stack on top of each other, they're going to be solid at room temperature, right? Like the molecules are going to be closer together. And if the molecules are going to be closer together, they're going to form a solid. So it's something like butter. Versus my polyunsaturated fatty acids are usually oils. They're usually liquid. And if you can think about it, because they're not stacking as nicely on each other, the molecules aren't as close. And because the molecules aren't as close, it's going to form a liquid instead of a solid, right? So you can just think about it like that. But I just want you guys to remember and at least be able to cross out in an MCQ that if you see something, you should know that saturated fatty acids are the bad ones and that polyunsaturated fats are the good ones. Good-ish. I mean, obviously you want it in moderation. The other thing is that regarding, okay, so I told you that for hypertriglycerides, the management is reduce your carbohydrate intake versus for cholesterol, you reduced your saturated fatty acid intake. You also want to increase your dietary fiber. Regarding high cholesterol, specifically hypercholesterolemia, you want to increase your dietary fiber. And the reason this is that the dietary fiber is going to reduce the amount of cholesterol and bile salts that is being absorbed. I don't know why, but the dietary fiber is going to reduce the amount of cholesterol and bile salts that is being absorbed. And so if you're reducing the amount of cholesterol that's being absorbed, it's going to lower the already very high cholesterol levels in the blood. Because of triglyceride, think of carbohydrates. I'm reducing carbohydrates, and cholesterol, think of reducing saturated fatty acids, and think of increasing dietary fiber. And the rest I think is pretty intuitive. For obese patients, you want to do weight reduction diet, but the problem is that you can tell people to try to exercise more and that kind of stuff. That's not necessarily, a lot of people still won't do that, and I don't do that. And so sometimes we have to resort to drugs. And so now we're going to get into the 2 types of drugs that you need to know about. Okay, so this is just a recap because I wanted you guys to remember this before I go into statins. But just remember that cholesterol synthesis is because of this enzyme, HMG-CoA reductase. Okay, HMG-CoA reductase is the enzyme that is responsible for cholesterol synthesis. If I inhibit cholesterol synthesis, I'm going to get lower cholesterol levels in the blood. Okay, if I have lower cholesterol levels, not in the blood, I'm so sorry. HMG-CoA reductase means lower cholesterol synthesis, which means lower cholesterol in the cell. It's going to decrease the amount of cholesterol that's in the cell. Okay, this is going to lead to sort of a domino effect. Okay, so if you remember, if there's low cholesterol inside of the cell, my cell or my hepatocyte is going to start to think to itself, oh, I'm low in cholesterol. I need to increase my intake of cholesterol. And the way that it's going to do that is it's going to increase the low cholesterol, is going to signal to the DNA sequence that I need to increase LDL receptor. I need to increase LDL receptor synthesis. Okay, so again, no, we're doing the opposite. So I'm starting, okay, so don't think of, okay, so I'm not sure if you're asking, so somebody asked, how can we inhibit LDL receptors when we have higher cholesterol in the blood? Exactly. So when you think of statins, you have to think that we're giving statins to somebody who already has high cholesterol in the blood, right? So we need to lower their blood cholesterol. So what I'm telling you is that you inhibit HMG-CoA reductase, is going to lower the cholesterol that's inside of the cell, and if low cholesterol is going to increase. It's going to increase LDL receptors. I'm saying it's going to increase the expression of LDL receptors. Not going to decrease, it's going to increase the LDL receptors expression. Okay, low cholesterol inside of the cell is going to increase the expression of LDL receptors. If I have more LDL receptors, why increase? Okay, just think about it like, think about your cell. Okay, I have my hepatocyte. If I'm decreasing cholesterol, okay, let's do this step by step. I have my hepatocyte. If I'm inhibiting HMG-CoA reductase, is that going to increase or decrease the cholesterol inside the cell? Type in the chat. If I inhibit HMG-CoA reductase, if I inhibit cholesterol synthesis, is it going to increase or decrease cholesterol inside of the cell? It's going to decrease the cholesterol that's inside the cell, right? If there's less cholesterol that's inside of the cell, do you think that your hepatocyte will want to take in more or less LDL from the blood? Will it want to take an answer in the chat? Is it going to take more or less LDL from the blood? Yes, exactly. I want to take in more LDL from the blood. If I want to take in more LDL from the blood, then am I going to increase or decrease my LDL receptor expression? Am I going to increase or decrease my LDL receptor expression? Increase. Okay. So I think what's happening, okay, so you guys have that concept down. So I can understand that you guys understand the domino effect. I think what's happening over here is that we're mixing up the statin slide, which is kind of my fault because I used the exact same diagram, and this slide. This is normal homeostasis. When I was explaining this slide, I was explaining normal homeostasis, which is what happens when I take in cholesterol, and I was saying the opposite of what I was saying with statins. With normal homeostasis, I was explaining the mechanism of what happens when we have high cholesterol inside of the cell and how there's a negative feedback inhibition to make sure that the cholesterol doesn't get too high. So that's what I was trying to explain over here. Yeah, sorry, it was my fault. I used the exact same diagram and I didn't clarify. Versus when I'm talking about statins, statins are a medication that I give only when somebody's cholesterol is already very high. Okay, so when somebody has already very high cholesterol, what I want, that's what I was trying to explain over here. And if they already have very high cholesterol, then one of the mechanisms that we can do is we can inhibit this enzyme over here, HMG-CoA reductase. If I inhibit this enzyme, it leads to low cholesterol levels. So if you remember when I was talking about homeostasis, I talked about the effects of high cholesterol levels. That's what I was explaining on the homeostasis slide. Over here, I'm talking about the effects of low cholesterol levels. If I have low cholesterol levels, it's going to increase LDL receptor expression. You guys told me that. You guys just told me that in the chat. If I have low cholesterol levels, it's going to lead to increased LDL receptor. So I have low cholesterol inside the cell. I want to bring in more LDL, right? So that I can bring that cholesterol back to a normal value. So if you think about this in terms of medication, if I think about this in terms of statins, the reason this is helpful is because all of the LDL that was over here in my blood is now being brought in to my hepatocyte. Because I'm increasing the expression of LDL receptors, that's going to lead to less LDL in the blood and more LDL in the cell. And really what that means is less cholesterol in the blood and more cholesterol in the cell. Okay, so I'm going to review this again in the next slide because it's pretty important. Okay, and I think in the previous one, I used a mnemonic, and I think it was more confusing than it was helpful with the SREBP2. I wasn't trying to make you guys remember the function of SREBP2. I was trying to help you guys remember the order of the letters, to be honest. Statins reduce everyone's blood pressure because I'm the kind of person that I get confused, like in exam, was it SREPB? Was it SERBP? So I need some sort of mnemonic that helps me remember SREBP. So just as a random mnemonic, I wanted to help you guys with statins reduce everyone's blood pressure, but this has nothing to do with the function of the protein. It's nothing to do with the function of the protein. I just want you guys to remember, I just gave you a phrase so that you guys would remember the order of the letters. But if it's not helping, that's okay. Okay, if the LDL receptor is low when you have high cholesterol. Okay, when you have high cholesterol inside the cell, make sure you differentiate between the two. Is it high cholesterol in the blood or high cholesterol inside the cell? According to the diagram, the HMG-CoA reductase won't work. Okay, wait, sorry, I have to read your question. If the LDL receptor is low when you have high cholesterol, okay, when you have high cholesterol in the cell, yes, the LDL receptor level is going to be low. It's going to lower it, yes. The HMG-CoA reductase will stop working. Yes, because if you already have high cholesterol inside the cell, you don't want to synthesize any more cholesterol. So yes, HMG-CoA reductase enzyme will be inhibited. So wouldn't the cell cholesterol is low? Yes, when the cholesterol inside of the cell is low, this makes more LDL receptors. When the cell cholesterol is low, this increases expression of LDL receptors. That is true. Is that what you're asking? Okay, yeah, that's true. If you have low cholesterol inside the cell, it's going to increase expression of LDL receptors. That's true. Because you want to bring in more LDL. Okay, so hopefully it makes sense now. So I told you that statins, they inhibit this specific enzyme called HMG-CoA reductase. If I don't have HMG-CoA reductase, I'm no longer making cholesterol inside of the cell. If I don't have cholesterol inside of the cell, the hepatocyte is going to think to itself, oh, I'm low on cholesterol. I need to increase my LDL receptor expression. So it's going to increase the amount of LDL receptors that are expressed. If I have more LDL receptors that are being expressed, it's going to bring the LDL from the bloodstream into my cell. If I'm bringing the LDL from the bloodstream into my cell, it's going to lower the concentration of LDL that's in my bloodstream, right? Because I'm moving the LDLs in my blood into my cell. So I'm removing the LDL from my blood. It's going to lower LDL concentration in the blood, and that's how statins lower cholesterol level. So it starts with this enzyme, and it's this domino effect. What was the name of the transcription factor that mediates that process that I talked about, the genetic process, the increasing or decreasing LDL receptors, like all that stuff? What was the name of that transcription factor? I have it on the slide if you look hard enough, actually. There's a name of a transcription factor. It's responsible for LDL receptor expression, HMG-CoA reductase expression. Okay, I only see one answer. It's SREBP2. Yes. SREBP2 is the name of the transcription factor that's used to used pretty much in these processes. Let me show you. This process, and I believe this process as well, but mostly this process, the LDL receptors. In order to affect the LDL receptor expression, you need the transcription factor called SREBP2. Okay, so the second medication, and I think we're almost done after this. The second medication is called cholestyramine, and pretty much what the cholestyramine is going to do. So remember in the beginning, I told you that the cholesterol from your liver is going to be secreted into bile acids. The bile acids are going to enter into the intestinal lumen, and they're going to be like reabsorbed through chylomicrons, right? So pretty much what cholestyramine is going to do is that it's going to bind to the bile acids that are not in my liver. It's going to bind to the bile acids that are in my GI tract. Okay, so I literally take this drug by mouth, I ingest it, and then the cholestyramine is inside of my GI tract, and it's going to bind to the bile acids. And it's going to tell the bile acids, hey, instead of being reabsorbed and going to the liver, I'm going to make you excreted through feces. I'm going to make you stay in the GI tract and be excreted through feces. So pretty much what it's doing is it's preventing the absorption of bile acids. Okay, and I told you that bile acids are made of cholesterol. And so pretty much what I'm doing is that I'm forcing the cholesterol that's inside of my bile acids to not return to my liver, to not return to my body, to my bloodstream. I'm telling the bile acids you have to be excreted and removed from the body and removed as stools. Okay, if you remember in my very early mnemonic, I actually had a sun next to the proposal. Oh, the meeting will close after 5 minutes. Oopsies. Okay, I think we're almost done. We can probably just create a new link if that happens, right? We'll see if they'll extend it. But if that happens, we'll just create a new link. But what I was trying to say is that, like, what I wanted you guys to remember with cholesterol, with the proposal mnemonic, that the proposal was happening with a sun, and it was a really sunny day. The reason I wanted you to remember that is because the sun should remind you of vitamin D, and that should remind you that cholesterol is involved in vitamin D synthesis. We will revise the receptors and the apolipoproteins at the end. Both of those things will revise. But what I just want you guys to understand is that it's like if I'm removing the cholesterol from my liver, the liver is forced to convert the cholesterol that's being stored into bile acids. Like, it's forced to convert more of them into bile acids. So it can't recycle them anymore, right? Like if I take the drug cholestyramine, the bile acids aren't going to be reabsorbed anymore, and they're not going to be recycled. And so pretty much what I'm doing is that because the bile acids can't be recycled, the cholesterol that's inside of my liver is now, it's not going to stay cholesterol. It's going to have to be depleted to make more and more bile acids. Before, I could just recycle my bile acids. It wouldn't be a problem. But now that I'm removing the bile acids from my body, I don't have the option of recycling them anymore. I have to keep making new bile acids. And the way that I keep making new bile acids is by converting cholesterol using the 7-alpha hydroxylase enzyme. Wouldn't this medicine inhibit 11 beta hydroxylase? I'm not sure why that would happen. It might, but I'm not sure. My understanding is just that it binds to bile acids, and then that removes the bile acids from our body and prevents the recycling. So that's what they mean by depleting the cellular cholesterol pool. All they're trying to say is that I can't recycle the cholesterol anymore, and so now I have to keep trying to remove the cholesterol that's in my liver. Okay, so related to the previous topic that I talked about before, do you guys think that, actually, I don't know if I want to ask this because I feel like this is kind of complicated, but this was this was related to what we talked about before, which is like that if I am lowering my cholesterol levels in my blood, I'm going to increase the amount of LDL receptors, or like in order to lower the cholesterol levels in my blood, I have to increase the amount of LDL receptors. Because the more LDL receptors I have, the more LDL is being moved from my blood into my cell. Okay, so if you guys remember in the dietary slide, I told you that there's a specific kind of diet that will prevent cholesterol and bile acid absorption. What kind of diet is good for people with high cholesterol? Because it prevents cholesterol and bile acid absorption. Okay, I see one answer, two answers, yes, high fiber diets. Okay, this is it. Do we want to stop the meeting and create a new one in case this ends, because we're at 40 minutes? And then I'll go over, again, the apolipoproteins, and I'll also go over the receptors. But should we end this meeting and create a new one? Okay, so we can create a new link.